Hey guys, so today I will be discussing aquaculture in mummichogs for an ecological risk assessment of pesticide usage. Mummichogs are small killifish that reside in estuaries, marshes, and tidal creeks along the Atlantic coast and extend from the Gulf of St. Lawrence to the Gulf of Texas. These remarkable fish have been proven to be one of the most hardy and adaptable fish known. They're intriguing due to their ability to sustain high temperatures as warm as 34 Celsius for up to 63 minutes. A temperature surely most fish would not survive for such a long period of time. They also cope with temperature fluctuations from 6 Celsius to 35 Celsius. And that's just one environmental condition that demonstrates the hardiness of the mummichog species, making them an ideal aquaculture candidate because it would be pretty difficult to kill them. In addition, mummichogs have been found to be tolerable to pollution, which makes the PCB-contaminated Superfund site in our own local New Bedford Harbor the perfect home. Lastly, their eating habits make them a pretty easy species to aquaculture because they'll eat just about anything. Aquaculture in mummichogs would provide researchers first-hand discovery of probable hazards and implications prior to introducing the chemicals to the natural environment. Pesticides have a long history of persisting in the environment, causing a plethora of issues from environmental health to human health. Therefore, it is highly recommended that this approach of utilizing mummichogs as an indicator species of toxic pollutants for an ecological risk assessment be thoughtfully considered prior to releasing pesticides in the environment. And how do we know they can accomplish this? Well, according to a 2016 study, high genetic diversity in killifish seems to allow selection to act on existing variation, driving rapid adaptation to selective forces such as pollution. The next slides will discuss more studies that prove the adaptive nature of mummichogs and thus why they should be used for an ecological risk assessment as a means to prevent pollution in the future. Our first case study explored how mummichog populations have adapted to anthropogenic contaminants in embryos from populations that inhabit heavily polluted estuaries, showing heritable resistance to POPs, which are persistent organic pollutants. Such resistance is not found within embryos of uncontaminated regions. The results indicated that mummichogs indeed had unique haplotypes that significantly correlated with polluted or clean sites. According to the study, adaptation to anthropogenic pollutants is often associated with the survival of individuals with high tolerance phenotypes, and in the case of Fundulus, these adaptive responses to contaminants occurred rapidly. Therefore, this species would serve as an excellent aquaculture species because since they show tolerance to pollutants, you could essentially test pollutants on them before releasing them to the environment. The table to the right just shows the different PCB contamination among the sites that they examined. This study characterized 4 distinct environments that drive adaptive evolution of the mummichogs. Even in the most contaminated sites where the pollutants exceed lethal levels, the mummichogs still appear to thrive. Their evidence suggests that widespread adaptive variation in dioxin-like contaminant sensitivity supports the conclusion that genetic adaptation to dioxin-like chemicals occurs at sites that are not extraordinarily contaminated. That is, at concentrations of sediment PCBs equal or greater than concentrations correlated with probable ecological effects. In simpler terms, they learned that there is an evolutionary influence of contaminants and other types of anthropogenic stressors. The map here shows the sites tested, and as you can see on the graph to the right, the solid curves represent the populations that exhibit tolerance to pollutants, whereas the dashed lines indicate sensitive populations. Therefore, it's clear from looking at the graph that the sensitive populations that are not used to lethal levels of PCBs will be the ones to die first. This very recent study examined the effects of PCB153, dioxin-like PCB126, or a mixture of both, on CYP3A56 mRNA in killifish that were 2 generations removed from the wild that didn't have any exposure to PCBs. The response from the mummichogs in the study confirmed that NDL-PCB can in fact induce gene expression in the mummichog species. And looking at this figure, you can see the lack of effect from the PCB153 on mummichogs in New Bedford Harbor, indicating that mummichogs show resistance. Since their specimens are 2 generations removed from the wild, it further suggests that resistance to the chemicals is heritable. So here's a few key concepts I hope you take away from this presentation. and a key point that we should take advantage of this pollutant-adapted robust species to help keep our waters clean because their presence in certain regions is already indicative of toxic pollutant levels. And here are my references. Thanks for watching!